Science for everyone. Welcome to the core of molecular biology. Today, we'll explore the central dogma. This central theory, known as the Central Dogma, was proposed by Francis Crick, who elucidated the structure and function of DNA. The central dogma is a fundamental principle that explains how genetic information is transmitted within living organisms. This principle describes three key steps: replication, where DNA replicates itself to pass genetic information to the next generation; transcription, the process of copying genetic information from DNA into RNA; and translation, where the genetic information in RNA is decoded by ribosomes to synthesize a protein with a specific amino acid sequence. These three stages explain the flow of genetic information within an organism. Crick used the term dogma to mean a truth not yet proven. He also warned against the misconception that dogma implied an absolute, unchangeable truth. In the 1970s, research on RNA viruses and the discovery of reverse transcriptase by David Baltimore and Howard Temin showed that genetic information could also flow from RNA to DNA. This discovery was a significant challenge to the absolute belief in the central dogma. Crick readily accepted this new fact and soon revised his central theory, acknowledging the possibility of information flow from RNA to DNA. After the discovery of the DNA double helix, scientists strived to understand gene function. During this period, most research was centered on the central dogma. As a result, scientists' research interests were focused on the genetic information that was translated into protein. In the 1970s, while studying adenoviruses, Richard Roberts and Philip Sharp discovered that mRNA was shorter than the corresponding DNA. They found that the DNA sequence of a gene was divided into exons, which are translated into protein, and introns, which are not. This discovery revealed that genetic information is discontinuous, a pivotal event that reshaped molecular biology. The discovery of introns also showed scientists that only about 2% of the human genome is made into protein. At the time, scientists believed the remaining 98% of the human genome had no special function, so they called it junk DNA. This 98% was dismissed as mere evolutionary trash. However, since the 1990s, it's been gradually revealed that this junk DNA plays a vital role in biological processes. Of course, even before the 1990s, some researchers consistently proposed that DNA not translated into protein could still have an important biological function. In 1993, Victor Ambros and Gary Ruvkun discovered microRNA in Caenorhabditis elegans, demonstrating that RNAs that does not produce proteins can still regulate gene expression. Subsequently, scientists discovered a variety of non-coding RNAs derived from junk DNA. Some, known as housekeeping non-coding RNAs, were found to be constantly transcribed in all cells to maintain fundamental cellular functions. Other types, called regulatory non-coding RNAs, act like a conductor orchestrating an orchestra, regulating gene expression based on a cell's state or environment, and thereby determining its fate. These discoveries showed that RNA is not just a simple intermediary, but a key player that regulates the entire flow of information between DNA and proteins. Consequently, the central dogma was expanded into a more layered and flexible theory that includes the functions of RNA. The central dogma serves as the starting point for understanding the flow of genetic information, and as one of the most powerful fundamental principles for explaining life itself. It remains at the very heart of molecular biology. Our scientific exploration continues. See you next time.